So I want to just begin by thanking the organizers for this opportunity to be with you today. It's really wonderful and great honor to address this esteemed audience, and I want to thank all of you for participating in this session. So I want to begin by stating that I have no financial interest related to the subject matter of my presentation. And what I'd like to do is talk to you about the IARC monographs process, and these are really done by independent experts, and I'll explain some of our processes with regard to that. I'd then like to focus on the key characteristics of carcinogens. This is a really new paradigm that we've been using at IARC, and I'll talk to you about how they can be applied in cancer hazard identification. And then I'd like to finish with to explain to you our process for evidence synthesis and how we classify carcinogens in the IARC monographs. So as Martin said, IARC, the International Agency for Research on Cancer, is the world's cancer institute. We are part of the World Health Organization, and we're a scientific organization that's really engaged in two types of activities. First of all, we generate data through interdisciplinary research, and we also evaluate data. And we do this by convening global experts in their fields of expertise. And we have a number of publications, the WHO classification of tumors, the IARC handbooks of prevention, and a number of other statistics and other reports that we generate. But we also do the IARC monographs, and this is a program that's been going on for nearly 50 years at IARC. So you may wonder, if you had a big pile of data, such as shown up here, a number of publications, well, how do they get turned into these iconic orange covered books that are known as the IARC monographs? And really all the answers to that are in this short document, which is the preamble to the IARC monographs. It's been published in every volume since 1971, and it's been updated based on the advice of independent advisory groups periodically thereafter. And this was most recently done in January of 2019, and you can access it online at the link on this slide. And it gives everything from the procedural guidelines for how we select participants, manage conflicts of interest, involve stakeholders, and conduct our meetings. And it also gives the separate criteria that we use for reviewing the different evidence streams, human, experimental, animal, and the mechanistic evidence, as well as the decision process that we use in our overall evaluations. So the first question you may have is, who actually does these evaluations? Well, it's done by not me at IARC, but it's done by these working groups, and they are independent scientists who have been screened for conflict of interest, and it's really up to them to review the science and develop the evaluations. Now, you may wonder how we find these working group members. Typically, most of them are identified through searching the literature on the relevant topics. But we also have the opportunity, if you would like to nominate yourself or a loved one to join a working group, we do have a public process for doing so. Now, I am part of the IARC Secretariat for these meetings. It's not up to me to make the decisions, but I do assist the working group in all aspects of their evaluation, including all the coordinating functions and convening the meetings. Now, one of the hallmarks of the IARC is that we do invite other types of scientists to join the meeting, people with a scientific background, and they fall into three categories. We sometimes have invited specialists. These are scientists with relevant knowledge, but may have a competing interest. Often they contribute in the area of exposure. They may have had experience working with a particular agent and know how it's manufactured and produced, and that type of information is valuable for them to share at the meetings. Frequently, we have representatives of governments and health agencies, national and international, and they're always welcome to join our meetings as well. Finally, we can have observers. Typically, these are scientists with a competing interest, maybe from a vested interest or a company, and they are there to observe, and they are allowed in limited numbers, but they are not allowed to influence the outcomes. And none of these three latter categories I've described can draft any of the text or contribute to the evaluations. So what are the steps involved in this process of going from a pile of literature to the book? And really in the revised preamble, this is now laid out into these 5 steps. If you're involved in systematic reviews, you'll find them to be very familiar. The first step is, of course, identifying the relevant information, then screening, selecting, and organizing the studies. And then it's really the working group who does these steps 3 and 4, which is evaluating the quality of those studies, reporting on the study characteristics, and then this final step, and we'll come back to this a little later, how is the evidence synthesized into the overall evaluations? So how does this process come together in space and time? Well, we typically announce our meetings 1 year ahead. And at that point, we do post a number of opportunities for public engagement. So we have a call for experts, and all of this is announced well in advance of the evaluation meeting. So people can write to us and say, hey, I'd like to join that working group. And these have specific closing dates by which we'd need to hear from you, the request for observers, and the call for data, which is a little closer to the meeting. And in the interim, we announce a list of participants of everyone who would be in the room, all of those categories of participants that I showed you previously. Now, in the olden time, if you can remember back when we all used to get together to have scientific meetings, these evaluation meetings occurred in Lyon over an 8-day period. We're about to launch our 3rd remote meeting, and in the last 6 months, so it is possible to do it through the Zoom and other technology formats. So this process is going on even under the COVID crisis. Now, we communicate the results in a couple of ways. We usually have a short publication in the Lancet Oncology within a couple of weeks of the meeting, and then we publish the full monographs 9 to 12 months after the meeting. So some of the hallmarks is that really these meetings are open. They follow the transparent published methods that we have available on our website. All the meeting participants have full access to the data that are being evaluated. And again, you can access the results for free from our website after the evaluation has been done. So now I'd like to talk to you about the evidence that we consider, and it's really down to 3 lines of evidence. There's the cancer in humans, and these are your typical epidemiology studies with cancer as an outcome. We have cancer in experimental animals, and these are your traditional bioassays. And then we have the mechanisms. And we have another chapter in the monographs on exposure, and this is now in the revised preamble, the updated preamble, I should say, is really closer to the cancer in humans. So there's quite a lot of emphasis on the exposure quality considerations in those studies, bearing in mind that cancer has a very long lag period, or at least it can. So understanding what that actual exposure was and how well it's characterized can be very influential in considering the quality of those studies. But I would point out to you that the mechanistic data can be pivotal when the human data are not sufficient. And this has been true in the monographs since the 1990s, but there have been a number of challenges with respect to the mechanistic data that have been highlighted. So for the volume 100 of the IARC monographs, where we reviewed all the known human carcinogens, these challenges really came to the fore. And if you're not familiar with the agents, there's approximately 120 that are known to cause cancer. They really are a diverse set of agents. You've got things like metals. We know about some pharmaceutical agents. You can think about tobacco, smoking, asbestos. So it's really also radiation. I don't want to forget that one, that we know cause cancer. And what we noticed was during this review there really wasn't a method to search systematically. So this really goes to that first step of the systematic review process. Well, how are you going to identify that information if you can't really systematically search for it? And that is also a challenge in terms of bringing uniformity when you may be evaluating different types of agents, not only that they are different, but their mechanisms may be different. Another issue is that the mechanistic database is increasing in volume, and it's also increasing in complexity, the types of evidence that is available. And how can you really analyze this efficiently? How do you really go to those best quality studies and most influential studies given this challenge? And all of these really lead to the last one, which is really the biggest challenge in any systematic review, is how can you avoid bias towards a favored mechanism. So with this is background, and a number of us in the field, I would say, had been wrestling with this issue for a long time. And IARC convened a couple of expert workshops to really look into it in detail. And from this workshop came this concept of the key characteristics of human carcinogens. And this is really the brainchild of Martin Smith, who's a professor at University of California, Berkeley in the United States. And I've listed here a number of the publications that have come out on this topic. If you're interested in reading more about it, this 2016 publication was really a landmark publication that really launched this concept that these key characteristics are the chemical and biological properties of established human carcinogens, and therefore data on them can provide evidence of carcinogenicity. And most importantly, for those of us in the IARC monographs and who are trying to do systematic reviews, you can use them to assemble the data relevant to mechanisms without needing an a priori hypothesis of the mechanism and how the agent may cause cancer. Now, you can think about these as kind of a top 10 list of the properties of carcinogens. I have variously asked different people, if you sat at your desk right now and started to make your own top 10 list, it may not, I would assert, be so different from the one here. And just to orient you a little further to this concept, this is a really nice figure that was drafted by Lisa Reiswick. And what it shows is in the middle are the human carcinogens. And for instance, if we just go around the circle from the numbers, you can see ethylene oxide. This is an epoxide. If you're a chemist or chemical toxicologist, you'll recognize that this type of moiety makes this electrophilic, and electrophilic agents can bind to DNA, be genotoxic. They can also affect DNA repair and induce epigenetic alterations. But we know about other types of carcinogens. We have these little drugs here sitting over by key characteristic number 7. Immunosuppressive drugs cause cancer. This is what they do in their day job. They suppress the immune system, but it turns out at nighttime that's a bad thing to have happened, and you can get cancer. Now, we've had a lot of news lately about viruses. There are viruses that actually do cause cancer, and they do this by really different, they have really different properties. So they cause immortalization of a cell, and they also do this 10th key characteristic, really all the processes that are involved in going from one cell to many, which we know that's how you get a tumor. Now in black, we can see some of the other carcinogens that we know. Some of the earliest ones identified, like benzene, we've known about for a long, long time, and it really has a number of these key characteristics. Cigarette smoking as well, and asbestos, and a lot of other fibers. So, and we just published this paper in Cancer Epidemiology, Biomarkers and Prevention, where we really have a very nice table showing, well, what are the endpoints that really relate to these key characteristics, and how can you use them? And we also, Martin Smith and Lauren Zeiss and I have launched a website where you can learn more about the key characteristics. one of the other speakers in our session, Laura Vandenberg, was involved in a project to assemble the key characteristics of endocrine disrupting compounds. So you can read about all these other related activities on this website. So, so why has this been so important for us? And it really comes down to the fact that what we do in our day job is really search the literature and then look at the results. And this is like the silver bullet that we had searched for a long time. So we can actually design a targeted search for an agent. It could be benzene, it could be any other carcinogen of interest that you may want to look for, and you can go down all the key characteristics, and then you can organize these. We use this program called Hawk, which is a freeware you can get onto this website, and it allows you to really show why you excluded every study, and why for the studies that you included, how are they organized? And this has been a huge help to our working groups, especially when the data are voluminous. You can go immediately to see how many different types of studies are available on each of the key characteristics, whether those are studies in humans, whether they're in other species, what the endpoints are, and that's really, really very helpful. So one of the questions we often get is, well, what about adverse outcome pathways? And, are those helpful to you? And how do you use those in your work? And I just want to share a couple of slides that really kind of illustrate. This is a published adverse outcome pathway for benzene, going through these different steps. It all sounds very, very plausible. First, it's metabolically activated, then it's genotoxic, and then you have these other events, and finally leukemia. But when we actually did the systematic search using the key characteristics, the picture is actually much, much more complicated. The direction of these arrows is not always 100% clear, whether A causes B or B causes A. The bottom line is it's all there, and what we do is focus on what's in the boxes and not the arrows, because this is something we can actually pull together using our methods and going to the endpoints. And we published this picture in the EHP article that I talked about. So with that as background, I'd like to talk to you now about the evidence synthesis and the carcinogen classifications that we do. And here's a reminder of the 5 steps, and now we're coming to the end of it. So as I mentioned, we have our 3 different evidence streams, and within each of these, we ask our working groups to categorize them using these defined terms. And if you go into the preamble, you'll see what each of these actually means. In the update to the preamble, the mechanistic and other relevant data really got us a major overhaul, so that it used to be strong, moderate, or weak, but no one actually knew what those terms meant. So now we have a definition of limited inadequate that's really much more similar to the other and much more aligned with the other data streams. And the strong evidence, much of the review is focused on the key characteristics as I've described. We have another possible things that the working group will consider. Sometimes you have a mechanism in experimental animals that's determined not to be relevant to humans. And we have guidance on judging that, and that's still retained in this preamble. And additionally, there can be the possibility of a mechanistic class. So if recently we evaluated some aromatic amines, and by the way, we know aromatic amines cause cancer, and sometimes when you're evaluating a new aromatic amine, you say, hey, this belongs to this other mechanistic class that we already know, and we know these are carcinogens, and therefore they should be considered to be probably carcinogenic. So then the overall evaluations really involve bringing them together into these famous groups. And these numbers here indicate how many agents in the history of the program have been classified. As I mentioned, there's about 120 agents that are carcinogenic to humans. So, and in the last just couple of minutes, I just want to highlight for you what's new about this overall classification process. And I think one of the biggest changes is that this used to be a consideration of the first two evidence streams, and the mechanistic evidence really moved things up and down in what we called upgrades and downgrades. And now it's a single step integration where you're able to just read across the table. There's also a clarification that the group 2B is based on one stream of evidence, and this is a clarification that really mechanistic evidence alone, if it's strong, can be used to classify an agent as possibly carcinogenic. Another is that the group 2A is really based on two independent streams of evidence, and one of these needs to be in a human, at least a human cell system, primary human cells. And these combinations of the different evidence streams with the mechanistic evidence, again, playing a strong role in reaching these classifications. So, and just to think for a second about the future, we have to remain positive about the future. I keep telling people, and we and the monographs have a full agenda of work that's upcoming. We published this recently from the advisory group that we convened in March 2019. And I just want to highlight for you that for many of the agents with high priority, this is really based on the fact that there's new mechanistic evidence that has become available and that could really influence a new evaluation or an updated evaluation of an agent that IARC has considered previously. I do want to acknowledge our fabulous team that's in Lyon, and I also want to acknowledge with much gratitude our support from the National Cancer Institute, the US NIEHS as well, and the European Commission. And if we are able to once again convene working group meetings, we would invite you to come to Lyon. This is a picture not far from the IARC. And I want to thank you very much for your attention as well. You can come to our website and stay up to date there on all of our activities. So with that, I will conclude and I'll turn it back to Martin. So thank you very much for your attention.